Great, thank you, Megan, and thank you all for joining me to talk about diabetes today. It's obviously a topic that's of near and dear to all of our hearts. Nearly everyone we knows someone who's affected by diabetes, and you can see here statistics from 2007 show that the percent of people with diagnosed diabetes is of unprecedented proportions. And I highlight diagnosed because actually plenty of people, up to 40%, don't know they have diabetes. Not trying to scare anyone, but it's true, unfortunately. So I hope I don't need to convince you of the importance of finding new treatments for diabetes. With that said, I'll tell you a little bit about what we're going to talk about tonight. The first half of the talk will be a background on diabetes. We'll talk about the history, how it's diagnosed, what are the different types of diabetes, and the current treatments. I'm spending almost half the time on that part of the talk because I think it's really important that we're all speaking the common language and that we all understand the concepts. It's really the most important part to understanding the actual approaches we're taking, and it will become much easier to understand if we're all speaking the same language. After that, I'll talk briefly about some of the applications we're making with modern scientific techniques to understand more about diabetes and to come up with novel ways to even think about devising therapies. And then finally, I'll close out the talk talking about what my group is working on towards finding new therapies for type 1 diabetes. Again, my group focuses on type 1, and I'll talk about the different types in a moment. But I'm happy to talk about type 2, for example, during the questions in the end. So we'll start with the background. When I say history, I mean history. We're talking about 1500 BC. The Ebers papyrus was discovered in the 19th century by Georg Ebers in Egypt. And it was a medical text that described all sorts of ailments. And one reason we think that it described diabetes is they mentioned polyuria. Polyuria is a Greek word, poly for many, urea for urine. And it's really the most obvious manifestation of the disease where there's copious and frequent urination when it's in an undiagnosed state. I got a real kick out of reading about a proposed treatment for diabetes, which involved things like elderberry and milk and beer swill and green dates. I don't know if anyone's actually made that concoction in modern days and tried it. Maybe they are onto something. I don't know. So nothing really happened for the next 1,500 years. And in about 150 to 200 AD, Galen, who was a famous Greek physician to the Romans, and one of his colleagues, Aretaeus, started to learn more about diabetes, or at least observe it more. I'm not saying they really learned anything about it. And they were responsible for coining the term diabetes, which is a Greek word which means siphon. You might say, well, why siphon? Well, again, getting back to this idea of polyuria, they actually thought what was happening was that literally the body was melting into urine. So it's this liquefaction of flesh and bones to urine. And no one had a clue it had anything to do with sugar or glucose, much less the pancreas. So again, this was sort of a disease of the kidney or disease of the urine or melting back then. And patients died days to really months of diagnosis. So we really think they must have been describing type 1 diabetes at this point. Again, I'll get to the types in a moment. Fast forward again about another 1,500 years, and Thomas Willis in Oxford was the first to add mellitus to the name diabetes. And he did this because of the sweetness due to the excreted sugar in the urine. And you might ask yourself, well, how did he know it was sweet, Well, there are plenty of stories where, well, spilled urine collected more flies, and I like that story a little better than other possibilities. Which leads us then to the name that we use today, diabetes mellitus. And of course, there are actually other types of diabetes. I'm not going to get into that at all. There are rare forms of there are even rare forms of diabetes mellitus, which I'm not going to get into either, but there are such diabetes insipidus, et cetera, that we can discuss, after the talk if you'd like. So advances in the 19th century taught us more about the role of the pancreas in diabetes. Paul Langerhans was a graduate student when he described clumps of cells that he found in the pancreas during anatomy class. He didn't know what they were yet, but they were these clumps. They were always there. Unfortunately, he died young from tuberculosis, but in honor of him, some of his colleagues named these clumps the islets of Langerhans, and we call that today. We usually abbreviate it in the lab, and today I'll talk about islets, but just to know some of the history is important, I think. This is a typical islet that's actually from a human, and it's from, unfortunately, an organ donor who was generous enough to donate their organ to science. But we've stained this here. This is from our lab for the different two main hormones that are secreted by the islet, insulin, which is made by the beta cells, and glucagon made by alpha cells. And again, beta cells are going to be a main portion of the talk today. Now, getting to, that was just a description of the pancreas, just the anatomy. What's the role of the pancreas in diabetes? Well, Oskar Minkowski is a huge figure in diabetes lore and understanding the history of the field. And he and his colleague von Mering actually discovered by accident that if they removed the total pancreas from a dog, just within one day, it would start to urinate uncontrollably with huge volumes and frequencies. So that discovery told them that the pancreas is involved in diabetes. And now starting to think really scientifically, they actually implanted a small portion of the pancreas back under the skin of the dog, and were able to resolve the symptoms of the dog. And this really was the final proof. Well, there's some factor in the pancreas that's responsible for diabetes. What it is, no clue still, but we've narrowed it down to the pancreas. Again, we've narrowed it down to the pancreas. We know it's a factor. What's this factor? Well, it was named insulin due to the word insulae, which is Latin for islands of Langerhans. So we still didn't have any clue about what the chemical structure of insulin was. And in the 1920s, Banting, Best, McLeod, and Collett worked together at the University of Toronto, and this is an iconic painting of Banting and Best hard at work in their lab. And they worked to extract insulin from the dog pancreas. Again, dog. They finally succeeded with dog number 401, so this gives you a sense of how much work was actually required to remove the pancreas, do the chemical extractions, and so forth. Because this was such a deadly disease, the time between these events was very short. So in 1922, the very first human was treated with insulin. This was a 14-year-old boy in Toronto who would have died without the treatment, and managed to resolve his symptoms and live a normal life for a type 1 diabetic, which at the time was still quite short. In 1923, something of a ruckus was started because a Nobel Prize was awarded, but only to the more senior Banting and McLeod of the team, and so it was an immediately controversial decision. Everyone's like, hey, wait, they didn't do that alone. There were 4 of them. And it goes to show that not all decisions, in science or by Nobel and so forth, remembers everyone appropriately. But I would point out that there's a Banting and Best Institute in Toronto that studies diabetes, that is focused on diabetes. And Charles Best actually had the most successful career of the 4 anyway. So, what's a Nobel Prize at that point, right? Easy for me to say, right? So let's go back to the pancreas. Where is it? Let's dive in a little bit on anatomy and geography here. So this is a picture of the abdominal cavity, and if you could imagine, we have the stomach here that's laying in front of the pancreas. We've removed it so that you can see behind it. And the pancreas has a head portion, a body, and a tail, and the tail points into the spleen sort of like a catcher's mitt. But it lays across the back of your abdomen. And the role of the pancreas is twofold. The first, which is actually taking up over 90% of the bulk of the volume of the pancreas, is for digestion. So there are structures called acini, made up of exocrine cells that produce the digestive enzymes. And they produce those digestive enzymes and secrete them into these ducts. And the ducts enter the small intestine and work to break down fats, break down carbohydrates, and so forth when you eat a meal. The other 5 to 10% of the pancreatic volume is made up of these islets of Langerhans. We met Paul Langerhans very briefly recently. And they're involved in hormone secretion, primarily, like I said, insulin and glucagon, which are the 2 counteracting hormones involved in glucose metabolism. So type 1 diabetes, which I mentioned several times, we'll finally get to what is it? It's a disease of cellular deficiency, fundamentally. And the cellular deficiency is in particular the beta cells in the pancreas in these islets, and they're destroyed by the immune system. So the immune system somehow gets its wires crossed and says, beta cell, you are foreign. You're like a bacteria. I'm going to attack you and kill you. And as a result, by the time someone has had type 1 diabetes for 10 years or more, they've lost over 90% of the bulk of their beta cells, and they produce no insulin. So you might ask, well, so what if they don't produce any insulin? What role does insulin play? Well, insulin is a very, very key hormone in whole body metabolism. So it has many bad effects if you don't have insulin in your body. When you eat a meal and you have glucose in your bloodstream, the pancreas has special cells, well, actually the beta cells themselves, which senses the increase in glucose due to the meal. The beta cells then secrete enough insulin to get into the bloodstream to compensate for that. What insulin then does is talks to what we call the periphery, the other tissues in the body, things like the fat cells, liver, and muscle, and tells them to either inhibit the breakdown of fatty acids, to inhibit the production of glucose, or in the case of muscle, to take out that glucose. And so the muscle is actually the largest tissue that's involved in disposal of glucose during a meal. This is why people might have polyuria if they have diabetes. You have much, much higher glucose levels in your blood, and that osmotic strain allows you to secrete all of the water in your body, basically, with the glucose. As I mentioned, type 1 diabetes is fundamentally a problem of not having enough beta cells to produce any insulin. And as it turns out, type 2 diabetes is kind of a twofold or manifold, actually, disease, which involves not only insufficient secretion of insulin, but also an inability of the periphery to respond appropriately to whatever insulin is there. So you have a vicious cycle where your pancreas starts by secreting insulin. Your periphery might not respond so well, so your pancreas says, well, there must not be enough insulin in the blood, so I'll secrete more. And it keeps secreting more and more until actually you have elevated insulin levels. Well, eventually this can't go on forever, so then what you actually have is literally beta cell failure. Beta cells burn out. They can't secrete any more insulin. And so a type 2 diabetic will end up sort of looking like a type 1 diabetic in that respect. And this just illustrates that point here that beta cell failure is central to both diseases. It's not exactly the same cause of disease, but fundamentally ends up being the same problem. And you see here that if we graph plasma glucose levels in the blood versus the insulin secreted by the pancreas, a normal person who's non-diabetic and lean, with the blue line here, might have a linear increase. As you have more glucose, you secrete more insulin. Your beta cells are behaving appropriately. People that are obese but not yet diabetic might have even more secretion of insulin. And this is the effect I was talking about. Your periphery doesn't respond as well. Your pancreas might say, well, we just need more insulin. So you see that this curve is a little bit elevated over the normal curve. And then finally, during the transition to diabetes, the beta cells burn out, and they just can't produce enough insulin to compensate for the glucose levels. So even though you might have very high levels of glucose in your blood, you don't have enough insulin, nearly enough to compensate. Good. So that said, how do we diagnose diabetes? Again, I'm not a physician. I'm just telling you how we diagnose diabetes. It's mainly you would have symptoms that would suggest diabetes, polyuria, as I mentioned. There's another Greek word, polydipsia, and that means drinking too much. So you'd have someone who's thirsty for water, can't stop drinking, but then also urinates all the time. So that might suggest a diabetic state. With that, then, you might measure what blood glucose levels are. And they're usually expressed in terms of milligrams per deciliter of blood. Fasting levels here are shown here, where a normal person would have below 110 milligrams per deciliter. By fasting, I mean, say you have a doctor's appointment in the morning, So you wouldn't eat the 8 hours before going to the doctor. That would be a fasting level of glucose. Alternatively, you can also, if you have IGT, which here is impaired glucose tolerance, you might then go for a glucose challenge. So you would drink something like a Coke, which has incredible amounts of sugar, and then 2 hours later measure your blood glucose, and you can see that the span of what is normal is increased somewhat, but it's the same kind of idea that as you increase, you're going through a normal to a sort of pre-diabetic state to a frank diabetic state. Once you're diagnosed with diabetes, your therapies are somewhat limited. In the case of type 1 diabetes, really insulin treatment is the mainstay. It's required for life. We don't really have a very good alternative. And in this case, it's injected all the time. So patients are, pretty much ascribed to a life of checking blood glucose all the time, injecting an appropriate amount of insulin. And not only can it be painful, but it can be expensive because even just over 4 months, you go through so many different syringes that the health costs and the lost productivity are major strains on our economy even. One more experimental approach is to try islet transplantation. And in this case, again, you'd have an organ donor, and islets can be isolated from the pancreas. And once these are isolated, they can be put in a syringe, and they're actually injected into the liver itself, which is a little bit counterintuitive. So it's not a transplantation in the sense of a heart transplant where you know where it would go in the body, and a liver transplant, you know where you'd put it. Islets actually get injected into the liver tissue itself, and the islets kind of lodge in the veins themselves and are able to still sense and secrete insulin in response to glucose. Unfortunately, in this case, after about 5 years, almost all of the patients end up being dependent on insulin anyway, so it's not turning out to have the promise it did, say, 10 years ago. Now, for type 2 diabetes, we have more drugs, but similarly, where we're looking for a scalpel, we have sledgehammers still. So we have drugs that can enhance the periphery in the response to insulin. These are different thiazolidinediones. You might have heard of, well, Avandia actually falls into the category of thiazolidinedione, and if you've read anything in the papers about that one has some heart-related risks as well. Metformin, which is one of the really blockbuster drugs for type 2 diabetes. There are also drugs that can enhance insulin secretion, but it does it in a non-regulated way. So you have to make sure you eat enough when you take them, because otherwise you can actually become hypoglycemic. So if you have too much insulin, your blood glucose can get dangerously low. So all of these have some, caveats with them. They're not perfect. And so, having new and additional, maybe more of these scalpels that I'm talking about would be much more helpful to people with type 1 and 2 diabetes. Good, so that's the background. I hope you can wrap your heads around that and have a little bit more appreciation of where we've come over, over the millennia at this point. I'm now going to talk just briefly about some of the applications of the modern scientific techniques that we've developed here at the Broad and at other places to approach and understand diabetes and learn new ways that we might treat it. One way might be to understand what genes are involved in the disease, what genes are associated with the disease. It's clear that, both type 1 and type 2 have very, very strong genetic components to them. But we don't really have a great understanding of exactly what genes those are. So things like genome-wide association studies, or GWAS, are run, and many of them have been run here at the Broad. In this case, you have a large group of patients, in this case diabetics, and you'd have a large group of people that were non-patients, people that you've tested, they don't have diabetes. You can then collect their DNA pretty easily, and then you want to compare differences in what we call SNPs. I'll get to SNPs in one slide, to look for what SNPs are there in the genome. If you have a particular SNP that seems to be enriched in the patient population statistically over the non-patient population, then you might say, well, that's a region that might be of interest. It might have a gene that we're looking for. What's a SNP? A SNP is a single nucleotide polymorphism. So in the case of, for example, C, there might be a T instead. And so these happen on occasion. In the humans, it's about 1 in every 1,000 base pairs. But there are 3 billion base pairs in the human genome, so this turns into a large number. And some of these are common variants, so over 5% of the population. Some of the variants might be half and half. Half have CG, half have TA. In that case, you might say, well, is one associated with diabetes or not? Now, when this has been done with type 1 diabetes, the same exact kind of analysis, it's GWAS. What we end up with is many genes, and you're not supposed to read this, but you're supposed to see that we have many of these genes, and they're all involved in the immune system. And I guess this is not terribly surprising. You might say, well, it's really a disease of the immune system, but the effect is on the beta cell, right? So the problem is in the wiring of what you're calling a foreign cell. It just so happens that the foreign cell happens to be a beta cell. And in the case of type 2 diabetes, over the course of the last 10, 15 years, we've accumulated many different gene regions that might be involved in diabetes, in type 2 diabetes. Now, each one of these accounts for a very small percentage of an increase in risk. So all of them together pretty much account for 10% of the risk of diabetes. So it's not the case where you have one gene causing one disease. That's Mendelian. That's pretty easy to understand. This is a much more complex situation where we might have many different things, each contributing 1 or 2% of an increase in risk. We've now learned over the last couple of years that many of the genes in this region, many of the genes in these regions point to beta cell biology. So for example, there are known changes where insulin secretion is decreased in the patients that have one particular SNP in, say, TCF7L2 region. So this might say, well, maybe we should pay more attention to beta cell biology. And this also points out that even though, for example, I told you my group is trying to study ways to affect beta cell function and number to treat type 1 diabetes, it might very well be the case that if we find something, it can help type 2 diabetes as well, which would be very, very exciting, obviously. So the fundamental question we have is, can we use small molecules to increase beta cell number and function? Ideally, we'd love to make a small molecule that would work in vivo, in other words, in a human. So we're not trying to increase beta cell number and function in a dish, but rather make a drug that we could give to you that would increase your beta cell number and function in your pancreas. And I said, small molecules, what are they? Well, we've all probably learned about the central dogma of biology, where we're going from DNA transcribed to RNA, translated to proteins. Well, we're introducing small molecules, which are organic synthetic molecules that affect dynamic flow of information as opposed to inherited. So each of these small molecules might affect DNA function. Some of them might affect a protein to upregulate or downregulate it. It might affect transcription so that not enough RNA is made or too much RNA is made. But anyway, chemical biology is the use of these kind of small molecules to study living systems. Good. So finally, I'm going to talk about approaches that we're taking in the lab to find new treatments for type 1 diabetes. And we're doing this by trying, again, like I said, to affect the beta cell. There are many different approaches you might take. You might say, well, actually what you want to do is decrease the immune system so that they're not cross-wired to say the beta cell is foreign. That is definitely an approach that's worth thinking about and working on, but we've decided in this case to narrow our focus for now on the beta cell. And we take 3 different approaches. First is, can we make more beta cells? Can we go from 1 beta cell and turn it into 2 using a small molecule? The second way is, can we take another cell in a different part of the pancreas and turn it into a beta cell? In other words, tell it, well, you're no longer an alpha cell, you're going to make insulin for us, and just sort of, co-opt that cell. Finally, then we're looking for small molecules that can protect cells from apoptosis. And this is the programmed cell death that goes on during type 1 diabetes and probably in type 2 as well. And then I'm going to give you a few examples of what we're doing in the lab in each of these approaches. And that'll give you, I hope, a good idea of where we are and where we're looking to the future. So first, beta cell proliferation. What we do is we actually use human islets. Remember, this is a human islet from an organ donor. And we're actually looking for small molecules that can increase cell division in the beta cell. So what we first do is take these islets and we break them apart. It's called dissociation. So they start out as a sphere. We can treat them so that we end up in single cells. We can then seed them into each well of a 384 well plate. And this is actually a tissue culture dish where each well gets a certain number of cells and a different compound. And that's what allows us to test so many different compounds in a relatively quick time. What we're looking for then is cells that are both dividing and that are positive for insulin. We can stain for insulin. We have a marker of cell division that's green. And then we can stain for a DNA just to identify every cell in every well. And we can use our analysis tools to find a red cell that's also green. It turns out to be pretty simple when you think about it. However, the adult human beta cell really doesn't divide, it turns out. There have been many studies that have looked at unfortunately, youngsters who have died of any number of causes and looked at their pancreas and looked to see how many beta cells were dividing. And there was a very clear decrease over time. So as someone goes from a newborn to basically an adult, the beta cells more or less stop dividing. So that by the time you reach 18 or you might argue even 10, you have the number of beta cells that you're going to have for the rest of your life, which makes this pretty much a daunting task. But at the same time, we do have physiological examples where we think beta cell replication is occurring. And those two examples are pregnancy and obesity. So we actually have more data in humans on non-diabetic obesity than we do on pregnancy. And the beta cell mass, which is just sort of a measure of the total amount of space that the beta cells are taking up. If you don't have diabetes, you're following this black line here, and it's pretty much steady throughout adulthood and even adolescence. In the case of non-diabetic obesity, the beta cell mass increases to compensate for the increased mass of your body. And it's thought to be occurring because of beta cell replication. So what we're trying to do is look for different either genetic changes, hormonal changes, other factors that involved that are responsible for causing, in each of these cases, beta cells to divide. We have seen, at least in the lab, that if we overexpress some genes involved in the cell cycle in beta cells, we can induce their division. So this is just an image. This is the final merged image broken apart into its various components. So you see here the DNA, so that's a nucleus. This is our marker of division, and this is our measure of insulin. So even though we have what I would call 4 beta cells, looks like one of them is dividing, and that to us was very significant and very good news that we can at least see it in a dish. And so we're now on the hunt for small molecules that can recapture this event on their own. Good. The second approach, and this is the most challenging it turns out, is can we turn another cell type into an insulin-producing beta cell? This is the process that is transdifferentiation. So first, let's look at pancreatic development itself. We have an embryonic stem cell. It goes through a number of different changes through progenitors that either become a liver cell, for example. If we have a pancreatic progenitor, that makes all the different cells involved in the pancreas. The acinar tissue, this is the exocrine cells I was talking about earlier. Ductal cells, which are involved in conveying digestive enzymes. and the endocrine pancreas, in other words, the islets of Langerhans. And then from the endocrine progenitors, different factors are involved in differentiating these cells to the different components of the islet. So we have beta cells, alpha cells. There are a few other minor cell types that produce hormones that are still important for metabolism, but only make up maybe 2% of the islet. So we do have now examples in the literature of conversion of different cell types from one to the other. Some of you might have heard about, I don't even have it here, but anyway, the generation of iPS cells from various fibroblast states. So a more differentiated state going back, that's this here. So a beta cell, when you overexpress 4 genes, can become, well, it's not exactly an ES cell, it's a sort of like an ES cell. Alternatively, we have small molecules that can turn an ES cell into, for example, a pancreatic progenitor state. And so we're interested in different ways of achieving more insulin-producing beta cells. One project we have going on is to look at alpha cells. They're the most closely related cell type to the beta cell. So we thought maybe, okay, that might be the lowest barrier to changing cell types amongst each other. And 2 trainees in the lab have performed a screen and found one compound that can induce, I think you can see that it's somewhat green here. This is an alpha cell that normally does not express any insulin at all. When you treat with this compound over several days, you start to see a faint, smaller amount of insulin produced. This is a great step forward. This is obviously not as high expressing of insulin as a beta cell is yet, so we haven't found the holy grail of a compound that can actually switch one cell type to another, but at the same time it shows that maybe we can make some stepwise progress and find combinations of small molecules that can induce insulin expression in an alpha cell. Another way of making more beta cells is from exocrine tissue itself. And I show here a blue arrow, which indicates that this has been done in the literature. And in 2008, there was a very big story out because a group over at Harvard found that you could, a combination of 3 different genes, PDX1, neurogenin 3, and MAFA. If you inject those into a mouse pancreas, after several weeks, you start to see these red ectopic insulin-positive cells in the rest of the mouse pancreas. And this suggests that some of the other exocrine cells in the pancreas are turning into insulin-producing beta cells. And actually, these mice are actually cured of an experimentally induced diabetes in this case. So it's very exciting because it's a true proof of principle that if we can find a small molecule that can do this, we can probably cure, well, at least we can cure diabetes in mice. We have found, Yuan is a graduate student in the lab, that these 3 genes actually work in a petri dish as well in human cells. So she took a pancreatic ductal carcinoma cell line, and this is, I call it exocrine-like. It's not exactly an exocrine cell, but it's sort of, it's close to it. In cell culture, she overexpressed all 3 of the same genes, and then let them sit in cell culture for 2 weeks. Change the media, you feed them, you keep them nice. And actually, the cells start to acquire many of the characteristics of beta cells, including the production of insulin. So these red cells have now formed from these blue cells, so suggesting at least they make insulin. It's not to say they're fully beta cells yet, as indicated by the beta-like, but it does suggest, again, that we can make some transitions, and what we need to do now is study more carefully what exactly is this transition. What are we missing to actually make them fully beta cells? And in order to do that, we're looking for small molecules that can upregulate the gene expression of those 3 master regulator genes that I just mentioned. So we've worked out a PCR-based assay where each line here represents a compound treatment. And so we're looking for lines that would be going in the left direction, which would be activators of a particular gene. And now we're in the process of running that screen to find small molecules that can upregulate one or more of those genes. The last approach is to try to protect beta cells. So even though they might be being attacked by the immune system, can we find a small molecule that allows the beta cells to say, hey, we don't care, we're going to stay alive and ward off the immune system? So a very, very simplified scheme of what inflammation is going on in the beta cell during type 1 diabetes. These inflammatory cytokines, so this interferon gamma, IL-1 beta, TNF alpha, you know, they're acronyms, they're important cytokines to us, and in combination, they work together to affect cell death in the beta cell. So what we found is that in a rat cell line, if we treat with these cytokines, the ATP level, which basically correlates with the cell number, goes down about 2-fold. And this indicates that the cells are dying, which is good. That's what we want to see because that's what's happening in the body. And therefore, a screening positive, a small molecule that we discover through screening, would restore this signal that's lost during cytokine treatment itself and make the cells look like they're happy. We ran the screen here at the Broad with Patrick and Danny, who are both in the chemical biology group. And what I show here is some of the screening results. This is almost 30,000 compounds screened. And we have a whole host of controls here. And we have a positive control where we tested it many, many times. And you can see the different instances here. What we're looking for are compounds that score as well as, or maybe even better, than this positive control. And we actually found plenty of them. And I'm showing one here that's real interesting to us. This is a compound that's actually been synthesized here at the Broad. And it has this kind of what we call a skeleton. And there are 2 different areas which have many different chemical options. So we can hang different pieces of chemistry off here and figure out which one is the best for this activity. It's really, sorry about the line here, it might be a little hard to see, but this compound in particular increases ATP levels, and it has its half-max effect at about 4.9 micromolar, which in cell lines is, you know, it's not too bad. And it actually also lowers caspase activation. Caspase is a measure of apoptosis, so we want a compound that does exactly this, that raises ATP and lowers caspase. So that's great, we have one. Both of these EC50s, which measures where's the halfway point of how active it is, are not, you know, they're not anything to write home about, they're okay. So what Danny did was, well, I'm going to take this R1 and R2 position here, and I'm going to make a whole bunch of different analogs, because he's a talented chemist, and we have many talented chemists that all work here at the Broad. And he made 27 analogs. And we're in the process of testing them to find which one is the best. And we have indication, preliminary indication, that one of them is quite a bit better than the original hit itself. And this is exactly how we get to finding, you know, what's our optimal compound. And I just want to close out here by saying we've also increased our screening to over 300,000 compounds through the NIH's screening network, which one of the screening facilities is actually here at the Broad, so it's very convenient. And Patrick and the chemical biology platform here performed all 300,000 compounds, and it took almost 2,000 of those 384 well plates, which I showed you early on, and 6 billion cells in culture. So this took, you know, many weeks and many hours in the lab, but fortunately a lot of robotic help. And this is the data here where our positive controls are in yellow. DMSO wells are behind here, but they're pretty good at 0 each. And what we're looking for are compounds that can act just like the positive control, hopefully even better. Good, and so this is a summary of the 3 approaches that we're taking. I just wanted to reiterate, this is, you know, really the take-home message. We're trying to increase the division of beta cells, we're trying to turn other cells into beta cells, and we're trying to protect beta cells. And of course, none of this is done by anyone alone. There's a huge network of programs and platforms at the Broad Institute that all work together in a nice, happy family, and we work with other people at other schools as well. And we have funding from the JDRF, and from NIDDK, and from the Broad Institute. So thank you very much for listening, and I hope that you have some questions.
As one of the 40+ million people who have the label pre-diabetes, I'd like to know what are my chances of shedding that label.
Well, if I knew, I would try to patent that. Anyway, I think that there's a lot of literature evidence that exercise has a very tremendous and short-term effect on mitochondrial function in cells. And this can help a lot in terms of insulin secretion. Insulin secretion is very, I didn't even get into this, but is very dependent on mitochondrial activity. Muscle function is also very important for disposal of glucose that you eat as a meal. So I would say, I mean, I think personally, and again, this is not clinical advice or anything, is that I think exercise is the most important thing. I'll just say that. Oh.
Hello. So what is are you looking for treatments essentially for only advanced cases, or are you looking for things that might help when somebody is pre-diabetic?
I think that in each of the approaches, there's some element of what stage they would be most appropriate for. The last one, the protection of beta cells from apoptosis, is one I think that's most geared towards early stage. There are no therapies for preventing the progression from pre-diabetes to diabetes. We're hoping that that last one, if we can protect the beta cell, might be something where if we can catch it early, in other words, figure out who is pre-diabetic, that we could administer something and even prevent it, you know. Again, we're at the Broad not making drugs per se. We're making chemicals that we think are leads that would be important for the biology. The actual making of drugs is a whole complicated business that involves not just a petri dish, but you know, absorption and excretion, and we don't tackle that at all. So, yeah.
In terms of people with type 1, is there anything that suggests, as you were talking about catching it early, someone who has type 1 who is still producing a little insulin, whether they're honeymooning or whether they're, you know, I have type 1 and I've only been diagnosed for 4 months. Do these approaches, would they work earlier? For example, can I preserve my beta cell function?
Well, yeah, I would say yes. My thinking is that if we found something that would work in an early stage of beta cell apoptosis to prevent that from happening, that the whole community would rally behind looking for easy, quick biomarker tests that we could give to infants and toddlers to say who's starting to get type 1 diabetes. We're not working on that now, but, and it's a little bit of a leap of faith on my part, but I think that if we actually had a treatment, you know, that backfill would happen, yeah.
Hi, am I on? Okay, so you have this approach that you're doing to look at protecting the beta cells from the cytokines that are killing them in the first place. Is it understood why that's happening? Are there greater numbers of the cytokines in individuals, or are the beta cells more susceptible in those individuals?
No, that's a great question. Those cytokines are actually produced by the immune system cells. So what happens is that the immune system, once it gets the alarm that the beta cells should be attacked, right, goes closer to the islets. And once they get closer to the islets, secretes an onslaught of the cytokines. So the concentration near the beta cells is very high. I don't think that, I don't think that there's inherently something about diabetic beta cells that's more susceptible, but I'm not exactly sure about that either. Yeah.
All right, thank you.
Sure.
This is probably more type 2 related, but the gastric bypass, people that have that, and then they obviously don't, their diabetes goes away, or
it's amazing, yeah.
Can you explain how that happens?
I mean, it's one of the most amazing and fascinating stories. So, and if you don't know, people who get gastric bypass surgery, before even the weight is lost, something within 5 days, are cured of diabetes. And so obviously it's not just from the weight, the mass itself. There's something about, oh yes, I'm sorry, type 2. Yeah, but I think most of the gastric bypass is, yeah, patients that have type 2 and gastric bypass have a complete cure of type 2 diabetes within something like a week. It's ridiculously fast, and it's amazing, but no one knows why. Yeah.
Why is the urine sweeter in people that have that type of diabetes?
That's a great question. It's mostly because it's supposed to be used by muscle and fat. Once you eat, you have high glucose in the blood, and that should go into the cells that are in your muscle. But they only do that in response to insulin. So if you don't have the insulin around, the muscle doesn't know that there's glucose or there's sugar around. So it just passes right through, basically. Yeah.
Is there any thought about some way to convince the immune system that it should stop attacking the beta cells?
Yes, absolutely. We're involved in another project that's very early. We haven't even started anything. We're just sort of planning out what would be the most beneficial in terms of targeting the immune system. And it's very difficult because anytime you attack the immune system with a small molecule, anytime there have been clinical trials, you induce, all sorts of susceptibility to infection and no one really, everyone's kind of scared of attacking the immune system with small molecules.
Is there only one type of type 1 diabetes? Are there any other reasons why beta cells could be destroyed? Right.
No,
why does it occur at
you're
different times for different people?
Yes, no, you're absolutely right. So there's actually a type 1A. So everything I've been talking about, technically, with some people in the field, is called type 1A. Type 1B is more of a late onset injury to the beta cell, and I'm really sorry, I can't remember exactly how that was caused. There are other types of diabetes, though, which are called MODY, M-O-D-Y, and it's called maturity onset diabetes of the young. So it's not exactly type 1 and it's not exactly type 2, but these are diseases where there's a specific mutation. Most of them are known what the mutation is, and they're pretty rare, but it's usually one gene that causes the disease. Yeah.
I know you're working on convincing other cells in pancreas to produce insulin, but is there a possibility, and I'm sure people are working on it, but if you can talk about it a little more about stem cells, to use them to compensate for the beta
Absolutely. People are working on that, people other than me are working on that. That's mostly in an in vitro setting, though. So if you took an embryonic stem cell, you'd have to take it in a dish and coax it to become a beta cell. There are plenty of people who are working on that, just that alone. Can we make a beta cell reliably out of an ES cell? It turns out to be pretty hard because an ES cell wants to become everything that you are, of course, because it's an ES cell. So you have to really force it. No, you're only going to be a beta cell. And it turns out to be really hard. If you happen to succeed, though, you still have to transplant those cells into the human. And so there are some caveats with that approach as well, because the cells get attacked and you have to worry about immune regulation of compatibility of donors, things like that. But it's definitely being worked on. Yeah, it's a good question.
Could I ask you to look into your crystal ball?
Sure.
All of these approaches have great possibilities. The question on the mind of most of us who are dealing with type 1 diabetes, of course, is how long? Mm-hmm. If you had to guess, and I know it's a guess, it's a forecast,
yeah,
but you're in the field, you're educated. If you had to guess, what would you think as far as a commercially, medically available, essentially cure for type 1 diabetes? Yeah. What do you think?
the last approach I talked about, the protection of beta cells, turns out to be, at least in our hands, the fastest moving. We actually have we have some really high value compounds already that we're ready to put into mice. And we've only been working on this for about 3 years. So I consider that pretty fast. The transdifferentiation approach, though, the hijacking other cells to become beta cells, it's long term, it's the most challenging, but I think if we can do it, mice have been cured of diabetes by doing exactly that, by telling the liver, you know, some of you liver cells are going to make insulin, and the mice are cured. I know it's mice, but you know, I think that has promise too. But I think the protection of the beta cells will yield the first fruit. I hope there will be more than one.
All right, well, with that, I think if folks have additional questions, you're more than welcome to come down and ask Bridget, and I would invite you guys to come to our reception, and also the next 2 weeks we have more lectures.
Thanks.